Hello, everyone. My name is Musaddiq Lodi, and in collaboration with my mentor, Dr. Bret, I am presenting to you gene rescue following frameshift mutation in endosymbiont bacteria, rare or common. This was research conducted at the Center for Biological Data Science at Virginia Commonwealth University in Richmond, Virginia. So here specifically, we are looking at endosymbiont bacteria in louse, and we're looking at frameshift mutations and their potential rescue functions of genes. So, let's start there. What is an endosymbiont bacteria? Well, endosymbiont bacteria are bacteria that have a mutually beneficial relationship with the host organism. So specifically here, we're looking at bacteria that live inside human head louse. And we're looking at different kinds of mutations and their potential rescue functions. So one kind of frameshift mutation we're looking at, one phenomena, is known as polyA tracts. PolyA tracts are a insertion of A bases inside a genome of an excess amount that can take a gene out of frame. So what does that mean? So you have a bunch of A bases that are inserted inside of a genome, and they might take the gene outside of its normal start and stop range. What that means is that the gene could potentially lose its function. So certain genes code for a different purpose, and if these A bases take it out of range, then it loses its purpose. In addition, there is an enzyme known as polymerase that also is beneficial to gene function. It's imperative for gene function to occur. And what we found rather oddly is this combination of an excess amount of A bases in combination with the poorly working polymerase enzyme actually leads to the gene function being rescued. Despite these two errors, the gene function stays the same. This is not something that you would normally expect to see. You can think of it as a two wrongs make a right kind of situation. So this has been studied in different kinds of endosymbiont bacteria, notably in fruit fly bacteria. And we want to put this to the test and see if it also happens in human head louse bacteria. So using different bioinformatic methods, using different gene prediction tools, gene alignment tools, we were able to analyze the genome of this human head louse. So out of 521 protein coding genes within this human head louse species, 97 of them contain polyA tracts greater than or equal to length 9. And when we align these genes along with orthologous genes inside related species, we saw that the alignments actually contained a lot of gaps. So what does that mean? Having alignments with gaps indicates that the bases don't necessarily line up correctly. And this is not something you tend to find in orthologous genes. Gene orthology is the idea that genes are conserved amongst related species. So if a species is related to another species and two genes are orthologous, it means that that gene evolved from a common ancestor. Since the genes are common, you would think that their alignments and their bases would be lined up the same. So that begs the question, why are alignments so problematic? And that could be for a couple of reasons, and it's why this research is still currently work in progress. So the problematic alignments may be due to the genes essentially not being in the same frame as predicted. So there are different gene prediction technologies, and when they predict where genes are, they do it based on start and stop codons. So if a gene is in a specified start and a gene is in a specified stop, then it calls that as a gene. However, since we are dealing with frameshift mutations, if these A bases get inserted and they shift the gene outside of the specified start stop, it might not actually classify it as a gene. So first we have to ascertain if our orthology is actually correct, if these genes that were called are actually orthologous. So if that's true, then what we will do from there is we'll actually zoom out of our genome. So we'll look at, not just in the start and stop region, but maybe a few hundred base pairs outside of these regions. And we'll align based on region and not by gene to see if, if the gene prediction technology did not predict the proper start-stop codon, we'll actually be able to zoom outside of that and analyze what a potential gene could be to determine if there's any orthology, if there is a phylogenetic relationship. If the orthology is incorrect, then what we'll do is we'll manually examine each alignment and see what the gene regions are. So by hand, we will go ahead and look at genomes and determine, where they're similar and what the genes would be in order to determine this orthology. And from there, we'll be able to determine if this phenomena happens inside human louse species, if this is a conserved phenomena, if it's common in this endosymbiont bacteria. This work was supported and funded by the VCU Undergraduate Research Opportunities Program. I would like to thank all my mentors and my family for support throughout the duration of this project. Thank you.